Hello, my name is Caron Harada, and my presentation is GPR160 inhibition for the mitigation of chemotherapy-induced chronic neuropathic pain. G-protein-coupled receptor 160 is a formerly orphan receptor expressed in multiple tissues in cancer. Its inhibition is associated with cell cycle arresting cancer cells. It is expressed in the dorsal horn of the spinal cord after injury. Inhibition of GPR160 with siRNA or interfering with its ligand, cocaine and amphetamine-regulated transcript peptide, reverses and attenuates neuropathic pain associated with nerve injury. We inhibited GPR160 in CCI, a nerve injury model whereby the sciatic nerve is chronically constricted. Intrathecal administration of siRNA against GPR160 both prevents and reverses mechanical sensitivity that is induced by nerve ligation. The y-axis indicates a measurement of mechanical sensitivity, expressed as the amount of force an animal will tolerate before exhibiting a pain response behavior, such as flicking or licking of the affected paw. Animals subjected to the same pain model, nerve injury, were sacrificed and spinal cords imaged using immunohistochemistry and RNA imaging techniques, showing increased GPR160 expression in the outer lamina of the dorsal horn of the spinal cord. This is a region of the spinal cord where we expect to see biochemical changes during chronic pain. Furthermore, GPR160 transcript expression was increased the most on microglia, a central nervous system cell heavily involved in inflammation and pain. Regarding GPR160's ligand, we identified the formerly orphan peptide CART, cocaine and amphetamine-regulated transcript peptide, as a ligand for GPR160. We did this by knocking down GPR160 expression in cell culture and measuring ERK phosphorylation after CART administration, and saw that CART could not activate cells that GPR160 had been inhibited in. Furthermore, inhibiting any target in the proposed pathway prevented CART-P signaling through GPR160 to cause hypersensitivity. We have established a role for GPR160 in CCI, but we wish to extend our findings to another model, CIPN. Chemotherapy-induced peripheral neuropathy is a major dose-limited side effect in patients receiving chemotherapy. There are currently no FDA-approved drugs specifically for CIPN, and it is treated to varying degrees of success with antidepressants, anticonvulsants, and analgesics. In a model of chemotherapy pain, siRNA targeting GPR160 prevented the development of oxaliplatin-associated pain. Animals were dosed with systemic oxaliplatin and spinal siRNA against GPR160 concurrently. The y-axis indicates mechanical sensitivity threshold again. Animals in red exhibit usual behavior for CIPN, with significant sensitivity appearing on day 11 and persisting through day 24. Animals with siRNA targeting GPR160 were prevented from developing this pain phenotype. In conclusion, GPR160 function is necessary for CART-P cellular activation. CART-P binds to GPR160 to activate the MAPK-ERK pathway and phosphorylate CREB, activating other processes in the spinal cord that lead to central sensitization. siRNA targeting GPR160 prevents oxaliplatin-associated CIPN, and GPR160 inhibition should not interfere with the anti-tumor activity of chemotherapy. With that, I would like to thank you, as well as my mentor, my lab, our collaborators, and our funding sources.